So, Friday, we are doing an animal behavior lab. If you are at home, you can do this too. You're just going to have to kind of make up your own choice chamber. And when I post the lab on probably Wednesday or Thursday, if you'll read through it, it'll make sense. But here's what I'm asking you to do as groups of 2. Bring in 10 roly-polies. Pill bugs, roly-poly, you know what I'm talking about? Okay, so if you don't find 10 by Friday or by Thursday as a group, then we'll need to figure something else out. Possibly we can use crickets, which I know we can always get because they sell crickets at the Chevron down there by Lowe's and all that. So, but roly-polies work well. They're not as jumpy and hard to maintain, but what we're going to be looking at is animal behavior. And so you're going to come up with an experiment, you're going to have a variable, and we're going to look at how that affects their behavior and what they do. Okay, so that should be an easy lab grade and give you a good idea of what a scientist might do when they're looking at an animal's behavior and what they're looking for. So don't forget about that for Friday. Pretty much every other day this week we're covering notes. Who are we missing? Matt. Yeah, I got you. All right, so in the notes today, we're going to go over part 1 that you were supposed to do on Friday, because I imagine even if you did this, you probably had a little hard time understanding these different components and parts and visualizing them. So I'm going to go back through part 1, and then we're going to hit what we can of part 2 here on how the nerves actually send a signal down the neuron. So on page 1062, we've got a picture of a neuron at the bottom of the page. But before we get to that, let's talk kind of basic overview of nervous systems. Obviously, we have a very, very complex nervous system with a lot of different components, and it's just there's a lot to it. But not all organisms are that way. So if you turn to 1080 real quick, flip over. This kind of gives you an evolutionary overview of the nervous systems in different organisms. Now, when we're talking about animals, plants can respond to their environment, but they don't have neurons like we have neurons. So these are organisms with neurons that make up part of that nervous system. One thing you don't see on here are peripherins, which are sponges. So no sense of any kind of nervous system there. But in the first ones and the most simple ones, here we go. Part A, read that for us, Katie.
Starts with the evolution of an organism's wide nerve net in jellyfish to help control movement.
Yeah, so cnidarians are the phyla of jellyfish, hydra, sea anemones, coral. These are all very similar organisms, and they have what's called a nerve net. So there is no central location for what we would call a brain or head or anything like that. They just have kind of a loose grouping of cells scattered throughout their bodies that helps them to respond to the environment. Okay, and you see a very similar thing in echinoderms and sea stars, where there's no head. It's not quite a net the way it's spread out like in the hydra, but does have some nerves that go out to the different body appendages so that it can react and conduct whatever activities that it's doing. All right, part B, Tay.
Evolution of the brain, a mass of neurons leads to greater control of the system. It utilizes a nerve cord to stand on.
Okay, brain. Sorry, it's Monday. A brain helps give you control over what's going on throughout the body. Okay, and that leads into part C, 5.
The evolution of other sensory organs in the head region called cephalization allows for reception and response to various types of stimuli from the environment.
All right, so cephalization means
development of sensory organs.
you got a head with some sensory stuff up there. So, like what we normally think of when we think of a head, like eyes or ears or taste or whatever. So that's cephalization. Now, you're going to see that term again probably like octopi and squid and that group. They're called cephalopods. Ceph means
Head.
head, and pod means an octopus has a head on top of a what?
Like,
Pod, feet, good. So, basically means head, foot, the same thing with the squid. And so, cephalization means you have a way of detecting stimuli in your environment, incorporating that into your brain, and you also have some sense organs in that region. So, that's what we see in most higher developed animals. All right, and you can see down through here how that kind of develops as you go toward vertebrates with these different types of nervous systems and other organisms. Okay? And kind of, we'll get down to we're mostly going to focus on humans here, though. All right, let's see. Overview of the nervous system. Turn to 1082. The sort of naked guy on the right side here. All right, overview of the sensory system. Sensory input. Mason.
Sending information into the brain or spinal cord, neurons in the body. Sensory receptors receive a stimulus from the environment. A stimulus is a form of energy such as electromagnetic light, mechanical pressure, or sound waves.
Okay, so we've got sensory input here. That's analogous to what?
I'll say reception.
Reception, good. So whatever is stimulating or causing you to receive some type of signal. Integration, Sarah?
This is the interpretation of the energy by the central nervous system, basically thinking about the stimulus. This interpretation of the stimulus leads to a determination of the appropriate response.
Okay, so integration would be like what?
Transduction.
Transduction. So the signal's being sent in, it's trying to figure out what it's going to tell us. And then motor output, Mariah?
Sending out impulses from the brain or spinal cord to glands or muscles to create a response. The response is carried out by effector cells. Effectors are muscles or glands. These structures can have an effect on the body.
Okay, so this is like your response. Good. It said response. All right, so motor output carried out by glands or muscles. These are called effector cells because they have some type of effect. What is your primary control system in your body? Your nervous system. Good. Secondary control system in your body is? Endocrine system, your hormones. So the nervous system controls the hormones, and that's where the glands come into place. It's talking about like endocrine glands. Muscles. Sometimes if you need to run, move, hide, whatever you're doing, muscles do that. And so there are neurons that go directly to those muscles, and they tell them to contract, to relax, whatever they're supposed to do. Okay? So that is in the motor output, the response area. Peripheral nervous system. So, before we go there, what is the central nervous system made of on page 1082? Brain and spinal cord. The PNS, the peripheral nervous system, what's it made of, Ashlyn? Sure.
Cranial nerves, ganglia outside, CNS, and spinal nerve.
Yeah, so it's made of all these peripheral nerves that branch off. I want to say there's 31 pairs off the spine. Maybe I'm wrong. It's been a while since anatomy class. But there's a bunch of peripheral nerves that branch off the spine, and then there's a bunch of cranial nerves that go out to different parts of your body as well. So that's kind of the basic overview of how the nervous system works. So there's some type of reception. The signal comes into remember, the signal is coming into the brain or spinal cord, and then there's a signal coming out as a response to somewhere out in the body, okay? Just keep that in mind. If you'll flip over to page 1070, let's see here. We're going to skip around a lot. Skip. No, I don't want to be on 70. Go to 62, sorry. The bottom of page 62, let's look at the basic anatomy of a neuron. And if I were you, I'd probably draw this out somewhere, and I would label this so that you're familiar with these different parts. In the nerve structure, the cell body, we call that the soma. This takes stimuli from different dendrites and compiles it into it compiles the energy into one signal like a funnel for fluids. So the cell body, it's that main portion of the neuron there where you find the nucleus and the ER and all the organelles and other cell parts. That's that main area. So signals are coming into that area and then they're sent off. It said signals are funneled into that area from multiple locations, and then signals are sent out the other side. Now we're going to look at this basic neuron structure on 1062. There are some different types of neurons. You can see on 1063 over here. We're not going to worry about those so much. You'll notice, well, just a kind of an overview, you've got basically sensory neurons that are detecting some type of sensation. You've got motor neurons that are sending out some kind of output to muscles or glands. And then you've got all these interneurons you see here, which are in between that are sending those signals to different locations. Okay? Well, we're going to look at this basic setup on 1062 here. Dendrites. Tell us about those, Miranda.
These collect and carry stimulus energy into the cell body. They cover a large area.
Okay, you need to know this. Dendrites carry signals in. It's not that hard to remember. Dendrites bring the signals in. Why are there multiple ones?
Because they need to draw in stuff from along. Like
They're gathering signals from multiple locations. So if you'll think about like all of the surfaces of your body, your skin and stuff, you've got all of these neurons. They're called proprioceptors, and there's some other kind of, they detect different types of pressure. And they're sending signals to your nervous system. So you don't have like a nerve for every one of those sensory cells on the outside of your body. You have like one that gets bigger and bigger as it goes into your central nervous system. And so what happens is those signals from all those different individual areas throughout your hands or skin or whatever you're talking about, they're funneled into a different neuron, and they're funneled into another neuron, and then they're kind of sent along that way. So it's funneled in so you don't have so much, I guess, traffic as you get up to your brain. So dendrites bringing signals into the cell body. Part C, what's his name? I forgot his name. It's Monday. Oh, please help me. Walker. I want to call you Hayden for some reason today. Sorry, bud.
Part C, what axon?
Dreyfus. Yeah, go.
This one arm carries the one compiled signal away towards the next neuron or effector cell.
Keep going.
The axon hillock. This is the
Hillock.
Hillock. Oh, sorry. This is the one swelling around the connection between the cell body and the axon.
Yeah, you'll see it's kind of large as the axon goes out. I don't know if this is mostly for structural support. I think it's what's going on here. But basically, the parts you need to know is dendrites bringing nervous signal into the cell body. The axon is sending the signal away. Axon away, den in. Y'all can remember that, okay? So it's sending the signal along. And you got to think about this directionally. So it's either a signal coming in from your periphery into your central nervous system, or it's a signal going from your central nervous system out to one of your muscles or glands, okay? As this is sent along. Page 1070. Sorry about that. The myelin sheath. How many of y'all watched Lorenzo's Oil in 9th grade? Almost everybody? Okay, good. I really like to show Lorenzo's Oil again, just because we do a lot of like biochemistry, talk about that, genetics, what's that called? Biosynthesis. I don't know if we'll get to it this year, but in Lorenzo's Oil, he had a genetic disorder where he didn't produce an enzyme, and basically what was happening is fats were not broken down that were supposed to be broken down. They were building up in his brain, and they were destroying the myelin sheath in his nerve cells. So neurons are a lot like electrical wire, okay? Or the axons are. And so electrical wire has to have insulation. Why?
So that it doesn't tear away at the structure.
Well, what happens if there's a cut in the insulation? What will the electrical do?
It'll go out.
Burn
It'll go out, it'll short it out, maybe burn your house down. So anyway, it shorts out, it's not going to send the electricity along. And so basically the same thing is happening here in the neuron. When you don't have this insulation around the axon here, then it's not going to send the signal along. And this part, this fatty part that's produced here is why we have, I'll try to cut open a brain for us at some point this week or the first of next week so you can see inside. But inside your brain, the inner portions is mostly made of white matter. And it's white matter because it's mostly like fatty tissues. There's a lot of axons where signals are being sent from one location to the other. And so it's like appears a whitish color because of all that fatty insulation. All right, let's see. Myelin sheath. Katie, read that for us.
This is a lipid layer of insulation around the axon created by Schwann cells.
All right, lipid. What is that?
Fat.
Yeah, basically fat in this case. Fat's good. Insulation. And so it works as good insulation. You know, if you're out playing in the snow or if you're trying to keep your signals going throughout your nervous system. Keep going.
It prevents the electrical energy of the neuron from burning the overlying muscle tissue. It is analogous to the rubber coating of electrical wires.
Okay, good. So and I want y'all to skip down to part 7 where it says oligodendrocytes and Schwann cells. You'll see that in the notes? Down at the bottom of the page. All right, so on 1070, they've got a picture of a Schwann cell here. I'm sure some guy named Schwann from who knows where figured this out. Tell us about Schwann cells, Mason.
This term refers to the insulating cells in the PNS.
What's the PNS?
Peripheral nervous system.
Okay, so Schwann cells are the insulating cells in the PNS. It acts as insulation around the axon. Okay. These cells produce myelin. Myelin is that lipid-based substance that's around the axon that helps to do the insulating. Okay. And if you skip up one to oligodendrocytes, what's a site? CYT. What does that mean? Cell. So it's a type of cell that refers to Schwann cells of the CNS. So oligodendrocytes are the cells in your central nervous system that produce the myelin sheath. So it's just two different types of cells, but they're both producing this myelin sheath. MS. Read that one for us, Miranda.
This disease is where the Schwann cells in CNS and PNS begin to die. The electricity traveling through the nerves then burns the muscles into permanent painful contractions until death.
Yeah, so that's problematic. You'll probably know somebody or heard of somebody with MS. And so there are a lot of, I can't name all of them, but the one ALD for sure, Lorenzo, MS, these all have to do with this fatty insulation that's being stripped away or it's breaking down. And then those nerve impulses are not conducting very well. And so that causes issues. Let's see here. Turn back time. If I could find a way. Okay, let's go back up. Okay, we talked about myelin sheath, talked about those. Go back up to synaptic terminal, part E. At the end of the axon, terminal means the end of it. So if you'll look on page 1075, it's probably the best picture in the book for this. No, hold on. 1072. Write that down. So on 1072, they've got a picture of the synapse. Now we're going to go in more detail later on, but today is just kind of an overview of this. Synapse is the, read that for us, 5.
This is a gap between neuron or between a neuron and effector cell.
What did we say about neurons the other day? They do not what? Touch. They do not touch other neurons. They do not touch effector cells. There's always a gap in between them. Okay? So these neurotransmitters, so if you've got a gap in between you and you're not directly touching for contact or for cell signaling, then you have to send some kind of signal over that gap. And you'll notice here on page 1072, we've got little signals that are going across the gap. We call those signals what?
Neurotransmitter.
Neurotransmitters. Sarah, read that for us.
transmit the signal across the gap. The most common neurotransmitter is acetylcholine.
All right, turn to 1075. Y'all don't have to memorize a lot of neurotransmitters, but acetylcholine is pretty important. We're going to talk about it when we get to muscle contractions, how your muscles actually contract. Remember, in order for your muscle to contract and to move, a signal has to go from your brain down your neurons all the way to wherever that muscle is, and there's a gap in between it, and so then chemical signals have to go across that gap into that muscle, and then it then triggers some response, which would be muscle contraction. Okay, so neurotransmitters help to transmit those signals, and if you look on 1075, you may recognize a few of these. We'll talk about GABA. We'll talk about norepinephrine. We've already talked about that. That's like related to epinephrine. Dopamine. Y'all probably heard that one. You know, a lot of people do dope. You people at home, you know what I'm talking about. I'm not saying y'all are doing dope, but never mind. So dopamine is a neurotransmitter that gives people certain feelings. Serotonin is another one y'all may have heard of. Nitric oxide, that's used for our friendly dentist visits where you get knocked out. Neuropeptides, there's usually an article I make you guys read for this unit out of National Geographic. I just don't know if we'll have time for this. But one of the people in the article is a neuroscientist, and her and her husband are talking about how, she's like, love is just an exchange of neuropeptides between certain cells in your brain. It gives you that feeling of love. It's not, anything magical like we think of love. Right, Walker?
Right.
So, but her husband was not happy with that. He's like, I don't think that's what love is. It's gotta be more than that. She's like, nope, it's just neuropeptides. Anyway, sounds like hard times in Marriage City at the neuroscientist's house. All right. So those are some of the different things that are used as neurotransmitters, and a lot of drugs work as neurotransmitters, either acting as a type of neurotransmitter or blocking some of those receptors that are there. All right, next thing, number 2, Tay.
The neurotransmitter is released from the postsynaptic cell.
All right, if you look on 1072, presynaptic is the cell before, postsynaptic is the cell after. So a neuron is sending a signal in one direction from like outside stimuli into the central nervous system, or it's sending a signal from the central nervous system to your muscles or glands. Okay, so presynaptic is the before the end of the neuron, postsynaptic is after that. So that signal has to be sent across in order for a nerve impulse to take place. Any questions? Yummy. All right, part 2. Flip over to couple pages, 1068. Now we're getting complicated. Okay. Important concepts. Number 2 and 3, read those for us, Ashlyn.
Establish concentration gradients from the source of energy. Active transport requires a transport protein in the cell membrane to the energy source.
What's a concentration gradient?
It's like when a substance is accumulated on one side and then it has to flow to a place where there's less of it.
All right, so you got a lot more of one substance on one side than you do another. So when you open up a membrane or some type of channel, then it's going to flow from high to low and help to do some type of work or action. So that's what we're going to see here. So all of this section here on the action potential and the graph and everything you see on 1068, this is all about how is a signal sent along from one neuron down that neuron. How is the signal sent? What constitutes enough stimuli in order to make a signal? Okay, make a neuron signal. Now, and keep this in mind. When I say neuron, we're talking about actual nerve cells that conduct signals throughout the body. There are other. Oh, hold on, I think I skipped it here. Sorry. Go back to part 1. There are other types of cells in your body. I did skip this. That help the nervous system to function. Okay? Sorry, I got ahead of myself. Turn to 1082 real quick. 1082. What do you call where those nerves come together right before they enter the spinal cord? You got that big bundle of nerves that are coming from a big region on the outside of your body. They're coming into the spinal cord. We call that ganglia. And there's a couple coming in each side that's where they integrate into the spinal cord. And then you've got nuclei, is a bundle of neurons in the CNS. So, like the brain is considered a collection of nuclei. Okay, and then there are numerous other accessory cells that are in the central nervous system throughout the body that help the neurons function properly. Usually when you have brain cancer, it's not because of neurons. Because neurons, you aren't expecting them to do what? Yeah, you're born with pretty much all the neurons, or most all the neurons you're going to have throughout your life. There may be new connections that form, new dendrites that are connecting to other cells. But in terms of new cells forming, that's probably not going to happen. Or at least not a lot that we know of. So, but you do have a lot of other cells that are working as framework around them and taking care of them. And so these other cells are accessory cells. So they're working around, they're helping the nervous system function. And a lot of times we call those glial cells, or one of the types is glial cells. Glial actually means glue, okay? So this literally you could translate as nerve glue. These are cells that are kind of helping hold the central nervous system together and help things function. And they're really important, but they divide, and if they divide out of control, what can happen? Cancer. Y'all may have heard of like gliomas. Glioma is a type of cancer of these glial cells that happen in the brain. Okay? All right, sorry about that. All right, let's go back to part 2 on page 1068. So, how is the signal sent along the neuron? Dun, dun, dun. All right, oh yeah, active transport requires transport protein. The brain uses a ton of energy because it's constantly having to reset these neurons and get them back where they can send a signal. So your brain and spinal cord uses a large portion of the energy that goes on in your body, especially when you're just resting or sitting around like this. I want to say it's like 65-70% of what your body's using. It's a ton of energy to do what these neurons have to do in order to function properly. Okay, so let's go through and look at this. Membrane potential. Read that for us, Sarah.
The ability for the membrane to potentially do work. This is created by electrical charge gradients across the membrane. Ion concentrations on each side of the membrane create the charge gradient. Negative ions inside, cations outside.
Okay, so ions just mean charged particles. Anion, the easy way to remember that is a negative ion. Cations, cats are so positive, blah, blah, blah. I don't know how you remember that. Okay, so let's be honest, cats are that weird. So anyway, you've got different types of ions. It creates a polarity or charge. So like if you had a bunch of positive ions on the inside and outside of a cell and you pumped a bunch of positive ones out, what's that going to do to the membrane potential inside the cell? So you started out, you had some positive outside, you had some positive inside, but you wanted to create a concentration gradient, so you pumped a bunch of the positive ones outside. The inside of the membrane is going to be what? It's going to get negative. That's right. So you created a concentration gradient that has a specific voltage because it's got these negative ions. You can actually measure the voltage here in a neuron to see what this potential is. Potential meaning the ability to do work. I need a drink. All right, so at rest, what's called resting potential. Let's read the notes for us there, Mariah, part A. Yeah, Na is sodium and K is potassium. So you might want to write this out beside there. We're going to call this the salty banana, okay? Salt for the sodium and banana for the potassium, since they're high in potassium, okay? So we're going to call this a salty banana, just to help you remember. Although I think potassium can form some salts, but we're not worried about that. All right, read that for us.
This is a cell membrane bound in protein. It controls the movement of the charged particles of sodium and potassium. Remember, these molecules can also be called ligands.
Okay, so there's a pump that helps to move these in and out in an effort to create a concentration gradient. Read 1 and 2 under that for us, Tay.
This function requires energy, ATP, or stimulus for work. This is an example of active transport.
It requires a ton of energy. Well, I don't know about a ton, but a lot. Keep going.
It is responsible for generating a nerve impulse.
Okay, so we've created this concentration gradient that helps a nerve impulse take place. So like I said, all this is about how does the signal send along. Gated ion channels. So there are a number of gated channels here where these channels like get a signal and they open up and stuff flows through them for a nerve impulse to happen. Chemically responds to chemically charged ligands. Voltage gated ones responds to the charge concentration. We're going to talk about both those, some examples, so don't worry about that too much. Let's get down to, let's go through the book here, probably easier first. On 1068, at resting state. Read that one for us, Ashlyn.
The gated sodium and potassium channels are closed. Ungated channels not shown maintain the resting potential.
Okay, so what's primarily on the outside? Thought there'd be a better picture of this. Sodium's primarily on the outside, and what's on the inside? Potassium. Now, what is the resting potential? Like, let's put an actual voltage on it. What's the number? 5? You'd have to look at the graph to figure this out. Page 1068. What is the voltage at rest? When a neuron is just sitting there, it's waiting to fire.
-75.
That's about -70, yeah. So negative, you can see a little bit better on the previous page, 1068 or 1067 if you want to. So resting potential has a negative voltage. Now, that's kind of hard to understand when you think, well, sodium's positive and potassium's positive. So how did it create a negative voltage? You've got to have what?
I know.
You gotta have more potassium than sodium on that.
Bless you. You've got to have more positive outside than you do inside. So you can have more positive outside, and you can have a bunch of positives outside and some positive inside, and then that creates a negative gradient because you've got more positive on the outside. So this is negative inside the neuron, a negative 70 millivolts. That's the potential there. Okay, so the cell has created this potential. Okay, number 2, Katie.
Depolarization.
All right, hold on a second. This polarization thing always used to get me, and it's confusing. When it says at resting state, you can think of it as polarized because it's down at -70. Okay, so it's like at one end. It's like down at the South Pole. Does that make sense? So it's away from like where it would just be at equilibrium, where it should be even out, like at 0. So it's polarized. It's at one end. Okay. Now, depolarization. Go.
The stimulus opens some sodium channels. Sodium inflow through those channels depolarizes the membrane. If the depolarization reaches the threshold, it triggers an action potential.
Okay. So give me a couple minutes here. When you're like a mosquito lands on the outside of your skin, if it touches it, it may touch it and it may stimulate it a little bit, but if you don't feel it, it's not enough. You didn't feel it because it wasn't enough stimulation to send a signal along. Does that make sense? But if it moves around enough where you can feel it, then that's because it stimulated that neuron to send a signal along and up to your brain. Okay? So if it gets past a certain point of stimuli, it reaches a threshold. The threshold is kind of like you get over the hump, essentially. And so this depolarization, Katie, means it's polarized, it's set at -70. Depolarized means it goes back the other way. So depolarization, it goes from -70 all the way up to the action potential, which is where? As long as it reaches the threshold. Once it reaches the threshold, that signal is sent along automatically. What is the action potential at what voltage?
Plus 30.
Yeah, if you look on page 1067, it's right at 35, a positive 35. So it depolarizes all the way to the opposite end, positive 35. And it did this. What flowed in?
Sodium ions.
Sodium ions flowed in. They were positive, so it made the inside of the membrane more positive. Does that make sense? Okay, good. So this is how your nervous system works. I know it's a little complicated, but y'all got this. So sodium ions are positive. They flow in, makes it positive. Reaches this action potential. That signal was sent along down the neuron. I feel you, Mr. Mosquito. Now, in order so we don't die in a lot of instances, that neuron needs to do what?
It's released.
It's got to reset. It's got to reset so it can send a signal again. Can't mess around, okay? So it's got to get that voltage back down, okay? So what's it do in step 4, Mason?
Following phase of the action potential. Most sodium channels become inactivated, blocking an inflow. Most potassium channels open, permitting outflow, which makes the inside of the cell negative again.
Okay, so a bunch of sodium flowed in, made it positive, but they needed to make it negative again, so the cell let the potassium flow out. And potassium's positive, so when it flowed out, it made the inside of the cell negative again. Okay? Now, the only problem is, in order for a cell to be back in resting state, what's got to be outside?
Positive.
Sodium. And what's got to be inside?
Potassium.
Potassium. Think salty banana. So then you've got all these pumps that are working, and then once it gets back down, lowered back to that negative 70, 75 volts, depending on, like, you can see there's a little bit of an overshoot there. Once it gets back down lower, then basically what's happening is these potassium-sodium pumps, they're pumping sodium out and they're bringing the potassium back in. So then they're pumping them back where they go. They reset in order for another signal to be sent. Okay? Y'all have a good day. It's so complicated. You guys got this.